have to make a disclaimer on this episode, but I don't think you can see it, to be fair. But I literally played netball yesterday, and I'm not joking. I was kind of like running to catch the ball, and I had my hands out in front of me, and the ball went through my hands and hit me like square in the face, literally
Oh my
like
god.
pinged off my face. It was the
No.
most embarrassing situation because I literally had my arms out in front of me. The ball went through my arms and literally ricocheted off my face as I was running.
Oh.
And I was just like, and then after I had like essentially the shape of a ball
I was
like
about to say, did
all
you have like
around
a ball
my
in your face?
eye and then like on my
No.
face. And
That reminds me of like being in primary school, like when the boys would be playing football and like you'd be trying to like run past and like you could hear them like the boots like whacking the ball and like oh my god, and like every now and then one of you would actually get hit in the face. And tell me why it was always on a freezing cold day and the ball was always wet and it fucking
yeah.
stung so bad. That's what it reminds me of. Taz, you poor little thing. Well, there's no
Yeah,
ball
and
imprint there
like
today.
obviously everyone on the court saw, so
It's
yeah, it was
tragic.
pretty embarrassing. But I it kind of wanted to make me ask you, have you obviously that wasn't a really major injury, but like have you ever like been injured or had something really embarrassing happen to you like in any sport that you've done?
That's a good question. I don't actually think so. I've done many sports in my life, especially when I was younger. My mum used to make me do like the most random shit. That's how I ended up doing karate. But babe, I was causing the injuries. I was a weapon in karate. So
I don't
you
doubt
know.
it, to be honest.
So actually, yeah, no, I've touched wood because I do some extreme sports.
I was
Hopefully, I
gonna
won't
say,
be
you never
injured.
fallen on your head on pole.
I mean, I have, but like that happens on a regular basis. Any of my pole girlies out there, yeah. But luckily, I've not really injured myself like badly. I mean, I've seen
Mm.
people injure themselves badly, and it's pretty bad. But yeah, what about you?
Honestly, I twist my ankle at netball every other week, so
You
probably
do
that's
actually. You're
probably
always injuring that poor ankle, like
it's meant
always.
to be a non-contact sport, but it's fucking brutal. These netball mums, they decide to like take it up as like a friendly, like as friendly matches with like other adults, and they're fucking brutal. These
Oh, for a
netball
second, I thought you
mums.
meant netball mums as in like they're on the sidelines. I was like, hun, you're like 30. Why are their mums watching it? Like, what the fuck?
No, I am playing the mums.
I
The
was
mums
gonna
decide
say.
they want to take it up as a sport because they watch their kids all the time. And they
Oh my
are
god.
literally so brutal. And it's just like, babe,
How can you be brutal in netball
Karen,
though? You're
babe,
not
can you
it,
just chill out? It's a non contact sport. Like, stop touching
yeah.
me, stop pushing me because I'll push you back and you'll break your hip or something. So chill out.
In reality, they're younger than us. The mums are younger than
Yeah,
us.
some of them look
Oh,
younger than me. I'm like, what are you doing to achieve this look? Because whatever it is, you need to tell me.
the peptides, babe. They're on the peptides.
Oh, don't even get me started about those. I
If you listen
feel
to
like
last week's episode, you
peptides are
know.
Bondi Central. Yeah. We covered them in last week's episode very briefly, but yeah.
Very briefly. That's all they're getting is a brief mention, shall we say.
Yeah.
That is all they are getting. I mean, maybe one day we will do an episode on them. Who knows? They could end up in the next season of Twisted. You never know what could happen on this show. But we will be talking about some other legitimate science today. So let's get into today's topic.
Welcome to the So Cultured podcast.
Where we explore science and tech unfiltered.
We're your hosts, Liv and Taz, two girls who love STEM and spilling the tea.
Each season, we take on a new theme, whether it's busting conspiracy theories, celebrating women in STEM, or breaking down the science in your favourite sci-fi films.
And of course, in every episode, we spill the laboratory,
where we share your wildest stories from the world of science.
Let's get into this week's episode of So Cultured.
What if you were your own twin? That is the topic that we are going to be covering in today's episode. So imagine being heavily pregnant, already looking after two children, and then finding yourself in a situation where you literally have to prove to the state that these children are actually yours. That was the position Lydia Fairchild was in Washington in 2002. She had separated from the father of her children and was applying for child support. And as part of that process, she was asked to do something that should have been completely routine, which was a DNA test to confirm maternity. The assumption behind a test like that is so basic that most of us never even question it. Of course, a mother will match her own children, and of course, DNA will confirm what everybody can already see in front of them. But when Lydia's results came back, they seemed to show the exact opposite. The children matched their father, but Lydia did not match the children she had carried and given birth to herself. At first, that sounds like the kind of thing that must be like a lab error. So the test was repeated, and then it just came back the same way again. Very quickly, this stopped being a strange result and started becoming a legal and medical crisis. Because if DNA is supposed to be the gold standard for biological identity, then what does it mean when a woman who has physically given birth to her children appears to not be their mother at all?
I don't think I've ever heard of this before. And yeah, like you say, that obviously has major implications for like even crime scenes, I would say. I don't even understand how that could happen. That's literally insane. Imagine like carrying a baby for 9 months, giving birth to it, and then a test comes back and it's like, yeah, the DNA doesn't match. It's not the yeah, what? How so was it a mistake or was it not a mistake? I don't understand how that's biologically possible.
No, 100%. This is exactly where your brain should go, Taz, because that's exactly where my brain went. Because as we know, DNA is supposed to be the one thing that is consistent across our entire body. And like you hinted at, it's what we use to confirm identity, prove biological relationships, and like you said, even solve crimes. So when a result like this comes back, it doesn't just challenge the situation, it challenges one of the most basic assumptions we have about how our bodies work. But what Lydia's case ended up revealing is that in some very rare situations, the assumption is not actually true. So before we even get into the weird part, we need to slow down and talk about why this case should have been impossible in the first place. So at the most basic level, DNA is the molecule that stores genetic information in your cells, and it's made up of 4 chemical bases, A, T, C, and G. And the order those bases appear in is what carries inherited biological information. But the part that really matters for this story is not just what DNA is, it's how we expect it to behave in the body. Now, under normal circumstances, you start as a single fertilized egg, and that one cell divides over and over again as development progresses. So almost all of the cells in your body end up carrying the same genome. So your skin cells, your blood cells, liver cells, reproductive cells, in theory, they have all come from the same original genetic blueprint.
Yeah, and I guess we've kind of touched on this before because we've spoken about epigenetics, because every cell basically has the same sort of instruction manual, but different tissues are reading different bits depending on what job they need to do. And that is essentially epigenetics, which, yeah, we've definitely spoken about on the podcast before. Liv, you of all people would know that because obviously that's what your PhD is in as well. So I find this case really intriguing.
Yeah, exactly. We have spoken about it quite a few times, like you said, but that is basically it, like what you just said. So the genome is the instruction manual, and then epigenetics is the layer that controls how that manual gets read. Obviously, that's a very oversimplified explanation, but it's accurate, so we can go with that. So a brain cell and a skin cell are working from the same underlying DNA sequence, but they are activating completely different sets of genes. And that assumption that one person has one consistent genome across their body is what a huge amount of modern genetics is built on. DNA gets used in different ways depending on the question you're asking, but all of those uses rely on the idea that the sample you take actually represents the person as a whole. So in medicine, for example, if you take a blood sample and look for a genetic variant, like something linked to a disease, you're assuming that what you see in that blood sample reflects the genome of the entire organism. In forensics, they tend to look at specific regions of DNA that vary a lot between individuals, things like short tandem repeats. And that variation is what allows them to match DNA from a crime scene to a specific person with very high confidence. Ancestry testing works slightly different, again, because it looks across loads of markers, often what we call single nucleotide polymorphisms, and compares your pattern of variation to large reference populations to estimate where your genetic ancestry comes from. And then like in Lydia's case, in parentage testing, you're asking a really direct inheritance question. So a child should inherit half of their genome from one parent and half from the other. So when you compare those markers, there should be a really clear pattern of overlap. So overall, like all of these methods are different, but they all rely on the same core idea, which is that your DNA is stable, consistent, and effectively acts as your biological identity. Now, to understand why that assumption can break down, we need to go right back to the very beginning and look at how a human body normally forms in the first place. So in the simplest case, reproduction starts with one egg from the mother and one sperm from the father, and each of those carries half of the genetic information. Now, when they fuse, their DNA combines to form a new genome, which is unique to that individual, unless obviously you are monozygotic identical twins, which we'll talk about in a second. That single fertilized cell then divides repeatedly, and as it divides, it copies that combined genome into every new cell. So you go from one cell to 2 to 4 to 8 and so on. And all of those cells are genetically identical because they all come from that same original fusion event. Now, sometimes development branches slightly from that standard pathway. So if that early embryo splits into two, you get identical twins. And because they come from the same original fertilized egg, they share essentially the same genome. On the other hand, if two separate eggs are fertilized by two separate sperm cells at the same time, you get fraternal twins. And in that case, you're developing two genetically distinct individuals side by side, each with their own separate genome formed from a different egg and sperm combination. So fraternal twins are kind of like genetically the same as siblings, for example.
Mm-hmm.
So most of the time, the rule is very clean. One fertilization event leads to one genome, which is then copied across the entire body. Even in the case of twins, you're still dealing with one genome per individual. But biology doesn't always follow the neatest possible pathway. And in very rare cases, something happens very early on in development that breaks that rule in a way that does explain cases like Lydia's. So if we go back to that stage of development where normally one fertilization event gives rise to one embryo, there is a very rare alternative pathway that can happen instead. And this is where biology gets really strange. So in some cases, two separate oocytes are fertilized by two separate sperm cells in the same cycle. So instead of one zygote forming, you initially get two. And that is actually the normal starting point for fraternal twins. And the important detail is that each embryo has its own independently assembled genome because each one comes from a different sperm and egg combination, right? If development proceeds as expected, these embryos, they stay separate and you get the two distinct siblings developing side by side. But in very rare cases, before tissues and organs are laid down, those two early embryos can fuse and continue developing as one individual. Now, when that happens, both cell populations are retained in the same developing body, and that means the resulting person is actually carrying two distinct genomic lineages derived from two independent fertilization events. And this is what we call tetragametic chimerism. And the name is very literal because tetragametic just means that four gametes, two eggs and two sperms, contributed to one body. And it's also why people sometimes use that dramatic line that you could have like a secret twin, but not in the way you think. Because what they're trying to describe in a pop science way is that what would have become fraternal twins actually developed as one person.
Yeah, I've genuinely never heard about this before. Like, I've never heard about like eggs from like two separate eggs fusing. I've obviously heard about one egg separating into two, but I've never heard about them fusing together before. So I guess like how common actually is this? Like, is it as common as, I don't know, like monozygotic twins where it like the egg has separated? Is it as common as that? Or
no, it's actually very, very rare. Well, it's considered rare, but it's also probably because it's also very underdiagnosed. So we don't have like a clean estimate in the way that like people always want. So we don't actually know like a specific number. And a lot of cases are only discovered accidentally during parentage testing or like blood typing. or transplant investigations, which means that there could absolutely be people living with it who would literally never find out, which is why people always say, like, you can have a secret twin and like never know, because a lot of people just don't even know that they have this, because in many cases there are no obvious signs at all. Now, there can be, in some cases, clues like patchy skin pigmentation, body asymmetry, different colored eyes is a really famous example. And by the way, I don't know what it is, but whenever I post a selfie on Instagram, I have this one guy who always messages me like, love your chimeric eyes. And I'm like, I don't know what you're talking about. Like, I've got the same colored eyes. But like that would be a good example of it, is like having different colored eyes, which I'm guessing you've probably seen of before. I don't know what that guy is seeing, but anyway.
Also, like, it would be extremely, extremely rare if your mum, like, if you, like, given that you're a twin, surely it would be like so rare if you both had twins as well.
Right. Like we're twins, and yeah. Anyway.
But I actually have like weird skin pigmentation on my side that's like the entire like one side of my body, and now I'm like, maybe I was a chimeric
I mean,
twin.
you never know. Like, there's so many causes for like hyperpigmentation or like skin pigmentation, but this is definitely one of the things that could cause that. So, yeah, not to scare everyone into thinking that you are your own twin, but yeah, other things that might be an indicator of it are like mixed blood cell populations, or in some cases reproductive or sex development differences, but none of those are universal. So the really important point is that chimerism can be biologically very significant without actually being obvious at all, which I just think is so interesting. And it's also why it often goes undetected until something forces people to look closely. And we're going to come back later to one of the best human examples of what that can look like in real life.
That is so interesting. Like, I never knew this as a possibility, and I also find it interesting that it is probably underdiagnosed just because, yeah, people just don't really have symptoms of it. It's like you ate your twin in the womb, but like actually it was at a cellular level.
Oh, I literally hear that phrase all the time. Like, I don't know if this was just me. Was this a universal experience that like every single one of us when we were kids like knew someone in our class who like would literally always tell everyone about how they ate their twin in the womb?
Yeah.
There was always one
There
kid.
was always one.
There was always
but
one
they were
kid.
definitely just lying, innit?
Oh, well,
In like primary
probably.
school.
We all lied when we were kids.
Yeah.
Another wild thing is that those two embryonic lineages don't necessarily contribute equally to every tissue. So if we think about it, development is all about lineage allocation, cell migration, and proliferation. So one lineage might dominate in the blood, while the other contributes more heavily to like skin, gonads, or other organs. And this is exactly why a DNA test can appear to contradict reality. The assay is not wrong, but it's just basically reporting the genotype of the cells in the sample it was given. So if you give it a blood sample, you're doing like a DNA test on the blood sample, it's obviously going to report back the genome that's in that blood sample, right? But the problem with that is that the sample may not be representative of the whole organism in the way that we normally assume. So in Lydia Fairchild's case, that is the key idea, because if the tissue being sampled carries one lineage, but the germline that gave rise to her children came from the other, then the biological relationship is real, even though the standard test appears to rule it out. And just to keep the genetics nice and sharp, this is also where we need to separate chimerism from mosaicism, because those two get mixed up all the time and they are not the same thing. So in mosaicism, you still begin with a single zygote and therefore a single original genome, but as development proceeds, mutations or chromosomal errors arise in certain cell lineages. So you end up with genetically distinct cell populations that all trace back to one fertilization event. But in chimerism, which is what we're talking about, the distinct cell populations originate from different zygotes entirely. So mosaicism is divergence within one genetic origin, whereas chimerism is coexistence of cells from more than one genetic origin.
This actually really reminds me about the aging, what if we didn't age episode, because we touched on epigenetics then, and I remember you saying in that sometimes you would look at the epigenetics and they would have reported themselves as a male, but then like what you could see on their epigenetics was, or like their DNA was female, and I'm like, what if they were
Or like XXY.
chimeras, you know?
That's so interesting because that's a really good point. I'd love to know like more about the epigenetics of like the two different, like how different are they? Well, they'd have to be very
Yeah.
different. Yeah, their epigenetic profiles would be like completely different because like one of the major epigenetic marks is DNA methylation, which only or like predominantly occurs on a cytosine base when it's sat next to a guanine base. So because the sequence would be completely different, then also your epigenetic profiles would surely be like quite different.
Yeah.
Depending on obviously
So,
what you're looking at, but yeah.
Interesting. Because like, yeah, I wonder if that's a way to like look to see whether it's like underreported or undiagnosed.
I
I
guess
don't
it's
know. Who
difficult because
knows?
you like say you're looking at so you've got like a blood cell and a liver cell, and the blood cell and the liver cell have different DNA patterns. Yeah. Epigenetics probably wouldn't be a good indicator because you would already, in people who don't have chimerism, you would already expect them to have different epigenetic patterns because they're different cell types.
Right.
Right? Because like a blood cell has a different epigenetic profile to a liver cell, and that's how it acts like a blood cell, and that's how the liver cell acts like a liver cell. Yeah, that's like differentiation.
Yeah, but I
So
was just
epigenetics would probably be the
meaning
worst
like because
indicator of
you
it.
could tell like if it was female. Usually, you could tell if a sample was from a female or male, but then sometimes they would report themselves as the opposite of what you're expecting. I wonder.
Yeah, or like most times they would appear as like some type of like sex chromosome aneuploidy, so they'd be like XXY rather than like XY, you know, or XX.
Yeah. Yeah, so I guess, yeah,
Interesting.
that is interesting. Can this happen with animals as well? Because I don't know if you've seen it, but on TikTok, there's these like chimera cats,
Oh my
and they're
god, the
like one
cat.
half of their face is like ginger, and then the other half of their face is like black or something.
Oh my god, yeah, I've seen them. They're literally the cutest things ever, and I feel like they always go viral on TikTok. Like if there's
Mm.
yeah, if I see any video of it, it's always got millions and millions and millions of views. And yeah, they are actually called chimeric cats because like that's what everybody calls them, because visually they do look like the perfect example of two different genetic identities in one, especially those ones like you said that have like this split face and they have different colored eyes. And in fairness, true chimeric cats do exist, but most of the famous tortoiseshell and calico cats people are often thinking of are actually mosaics, not chimeras. So their patchy color pattern usually comes from something called X chromosome inactivation, which interestingly is related to epigenetics. And that is a dosage compensation process in female mammals where one X chromosome is randomly silenced in each cell early in development. So if the coat color alleles differ between the two X chromosomes, that random inactivation creates visible patches where one allele is expressed in one clone of cells and a different allele is expressed in another. So people call them chimeras because the patterns literally looks like genetically split. But in most cases, the mechanism is actually mosaicism rather than true embryo fusion. And I actually think that makes the biology even more interesting because two animals can look similarly like patchy on the outside or split on the outside while getting there through completely different developmental routes.
Yeah, I feel like this is the thing I'm still stuck on, because I guess how would you even know? Because you're saying this can be completely hidden or related to other like DNA changes or like effects, like mosaicism, like you said. So is this something that people can ever actually measure or like see in their own body, or is it only ever picked up through like weird test results like Lydia's?
To be honest, you probably would only find out, like I said earlier, through testing. And I'm going to tell you two examples of that. So a really good example is a woman called Karen Keegan, who was actually in her 50s when doctors were preparing her for a kidney transplant. And as part of that process, they tested her DNA to see if her children would be like suitable donors. Obviously, they'd be like the first people that you go to, right? And what they found is that 2 of her sons did not genetically match her in the way you would expect for a biological parent. And obviously, after this episode so far, we know why that would be. But I just think that's mental. Like, imagine you literally are going for like a transplant, and it's not until you transplant that you actually find out that you are chimeric. Like, I just think that is absolutely crazy. But then on the other end of the spectrum, there are cases where you actually can see it in the body, and I guess that's where it becomes more of like a tangible thing. And a really well-known example, really famous example, is a woman called Taylor Moore, who is a model and singer. And she noticed from her young age that her body had a really clear line of pigmentation running down it, where one side of her torso is visibly different in color to the other. And that wasn't just a cosmetic difference, it reflects the underlying distribution of 2 different genetic lineages across her body. So, developmentally, what that suggests is that those 2 embryonic cell populations contributed to different regions during early growth, creating that kind of visible boundary. And what makes her case even more interesting is that it isn't just about appearance. She's also spoke about having immune related issues where her body can respond to cells from the other genetic lineages as if they are partially foreign. So that starts to bring in immunology as well, because now you are dealing with a system that evolved to distinguish self from non-self, operating in a body where self is not genetically uniform. And again, that is not something that happens in every single case, but it definitely shows you the range of ways in which chimerism can manifest, from being completely invisible to visibly distinct to physiologically or immunologically oh my god, I can never say this word, immunologically complex. So what that raises is the idea that chimerism can actually exist on a spectrum, and one of the most common forms of that is something called microchimerism. This is really cool. So during pregnancy, there's this really fascinating 2-way exchange of cells between the fetus and the mother. So fetal cells can cross the placenta and actually enter the mother's bloodstream, and maternal cells can also move into the fetus. And some of those cells do not just disappear after pregnancy, but they can actually persist for years and sometimes even decades. And these are not just passive cells either. They can actually integrate into different tissues. And so there are studies which show fetal cells in maternal organs like the liver, the lungs, actually even the brain as well. And in some cases, they actually behave a bit like stem cells, differentiating and becoming part of that tissue. So you can have a situation where, for example, a woman is carrying around a small population of cells that are genetically distinct from the rest of her body, which actually originated from a pregnancy that happened years earlier.
Yeah, I think I've actually heard of this before, because I feel like, unfortunately, obviously, when people get pregnant and then have a miscarriage, a lot of people say, like, well, you could be still carrying on your fetus's cells, or like a part of that like potential baby with you. So as like a nice kind of, I don't know, like you're still carrying them with you kind of situation. So I've definitely heard that before, but I obviously didn't know like how rooted in science it was and like how much of a legit thing it is. So yeah.
Yeah, it's a legit thing, and it has been detected like decades after pregnancy in some cases. And scientists are still trying to understand what those cells are actually doing, but there is some evidence suggesting that they might play a role in tissue repair or might contribute to healing processes. But there is also research linking microchimerism to autoimmune diseases, where the immune system might start reacting to those genetically distinct cells. So once again, like in Taylor's case, you have this situation where the boundary between self and non-self is not as clean as we like to think it is.
Yeah, this feels like, I've honestly never heard of this before, and it kind of makes you think like, I don't know, I could be a chimera and no one would ever know.
No one
And
would
kind of
know.
like weird how you get to autoimmune diseases as well, but that makes like total sense. I guess, yeah, we think about DNA as if it's this one fixed thing that defines you, but like obviously you've also mentioned microchimerism when you have a child. So yeah, they feels a lot more complicated than just like having one set of DNA.
No, 100%. And like you say, like, what if it is more common than we realize? You know, like, not in a dramatic way, and not to suggest that there is like loads of undiagnosed cases everywhere, but just in the sense that there may be people walking around with more than one set of DNA in their body who would literally never know, which is insane to think about, because nothing in their life might ever trigger the sort of testing that could reveal it. Like, you know, I'm 28. I've never had any kind of test in my life. Actually, as a lie, I actually have had a DNA test, because I think I've told this story before, but my mum wanted to see if me and my twin sister were identical, because everybody told her that we weren't, because we were in different sacks. So I have actually had a DNA test, and obviously we were identical. But there's so many people.
The same.
Huh?
You literally look the same.
You look the exact same. But that's why she was like, nah, they're identical. Like, but she was always told that we weren't. But yeah, I mean, think about it. There's so many cases, or so many people who would just like not have any of these tests, or like they could potentially go until like they're like 80, and just never find out. And in some cases, actually, I'm not going to say that. Also, might be a really dumb thing to say, but could you potentially get away with murder?
I was thinking that, because obviously, like, her DNA didn't match.
Yeah. So like
her child. So I'm like, if her skin cell DNA was like, and hair cell DNA was like completely different to.
Yeah. And then they took your blood to test your DNA, or they took saliva, because that's what they would do, right? They would take your saliva. So yeah, if you bled out at the scene and they took that DNA and then they did a saliva test, oh my God.
I
Just
guess
thinking about this, they should implement a new law, which is wherever they get the DNA from, wherever they think the DNA is from at the scene, that's what they should take it from you. So like, if they collect blood from the scene, they should test your blood. I know obviously it's very rare, but like,
yeah, true.
I could be convinced.
I don't think they would always. I don't think they would always know like where they're.
But they should take a
DNA from.
A best guess. Like, you're gonna.
Yeah. But you would appear as at least related, wouldn't you? Because it's like just a different proportion of genes, right?
This is a good question. Okay, so let's think about this. Separate. Well, it'd be a separate. Yeah, you would appear as related. Yeah. But then you could just be like, well, especially if it happened in like your house, then you could be like, well, several people who relate to me also live in this house, and now what? Well, me and my twin sister always say this. Like, we say, if we were to, unless we left fingerprints, we could potentially commit murder. Not that I'm saying I'm going to do this, but just for argument's sake, right? Okay, so say she commits murder, doesn't leave any fingerprints, and then she blames it on me, and then I'm blaming it on her, and we're both in court. We both have no alibi. We both have, like, they've got equal against both of us, and like, we're both blaming each other. Then what do they do?
Actually,
technically both of our DNA was found at the scene.
Who knows?
And no fingerprints to, you know, say who it was, because that's the only thing different about us is our fingerprints. Then what?
I don't know. Would they jail?
Do we get away with murder, or do we both get sent down for murder?
Don't know. Surely both.
No, but I didn't commit the fucking murder. She committed it. It's not my fault I've got the same DNA. That's so unfair.
Yeah, I actually don't know what would happen. I mean, it would be pretty rare to like have the same level of evidence against you both. But if that was the case, I actually have no idea.
But this is the thing. I reckon nowadays, I wonder if they could do epigenetic profiling on the DNA to then test which one of us it is. But
depends how much DNA you leave at the site, though, doesn't it?
Yep. And then also if it's like, if they could do epigenetic profiling on it. But then a really good defense lawyer would argue that, like, that's not good enough. Anyway, you could potentially get away with murder.
We're on a tangent now. Yeah.
You could potentially not. But anyway, it's a very interesting case of science, which I will also say I actually did not hear about until I started doing the research for this episode. So I've actually never heard about it. I've heard of chimerism, but I didn't know what it meant. And I definitely had not heard of tetragametic chimerism. So there you go, guys. If you hadn't heard about it, you know now. If you had heard about it,
Yeah.
let us know, because I definitely wasn't taught it again in my 3-year degree. So I think it's very interesting.
To be fair, we are coming up to having done the podcast longer than we were ever in undergrad. So
Oh
I
my
think
goodness, that's crazy.
at this point, we have been learning a lot on this
We've covered
podcast.
so many topics. I was saying to Taz the other day, guys, like when it comes to picking our topics now for our different seasons, like me and Taz will always discuss what topics we're going to do. And I feel like we've covered so many. Like I'll be like, oh, this will be really interesting for an episode. But we've kind of touched on that in this season. So I want to do something completely different. But like we've covered so many things now that it's hard to find topics that don't overlap with other episodes. But I feel like that's kind of nice because we're always back referencing other episodes and
Yeah.
nostalgic.
Yeah, I mean, this was totally different, I feel like, to anything we've done before. So thanks so much for that, Liv. And obviously, if
You're
you're
welcome.
enjoying this season, don't forget to give us a little thumbs up or leave a comment. But that is not the end of the episode. We have obviously got our laboratory, and it is hot off the press with a listener story. So let's get into
Let's
it.
go. This episode is sponsored by Cite, the smart citation tool for your everyday research needs. Taz, I don't know about you, but I think Cite really sets itself apart from other citation and reference tools.
Yeah, I feel the same. With Cite, not only do you have access to over 250 million papers, but it can summarize a paper into a report, format citations, and more.
Yeah, and not only that, but I also love that you can connect it to tools like ChatGPT, Claude, or Copilot, and it basically just upgrades how those tools actually handle research.
Yeah, so instead of getting a general answer, your AI is actually searching through a huge database of research papers. And the key thing is it's not just looking at the titles or abstracts, it's searching inside the full text.
Honestly, it's such a slay because it just makes such a big difference, because a lot of important details don't really show up in abstracts. And I feel like most tools just miss that completely.
On top of that is the smart citations feature, which I think is one of the most useful features because it doesn't just show how many times a paper's been cited, it also shows how it's been cited.
Yeah, you can see whether other researchers have supported the findings, contradicted them, or whether they've just mentioned them. So it gives you a much clearer picture of what other experts in the field feel about it.
It also helps with access. So if a paper is available to you, Cite will help you get the full text.
So if you want to try it, you can head to cite.ai, or you can find the link in today's episode or video description. Setup only takes a minute, and you can use the code on screen now for money off. And thank you so much to Cite for sponsoring today's episode. But for now, let's get back into it.
Okay, so the tagline of today's email says, "My entire PhD lab is sabotaging me." So it says, "I'm writing to share what I've been going through in the lab I'm currently in. I'm an international student on a visa in the US, and I started my PhD in 2021. So now I'm in my 5th year and close to graduation. When I joined the program, I was encouraged to work with this professor because someone recommended him, and after rotations, I joined his lab. At first, the professor invited me to contribute to what was supposed to be a shared project, but in reality, it became entirely my idea, and it was the project I defended during my comprehensive exam. It took me 2 years just to explain very, very basic concepts. The professor wanted to move in one direction while I wanted to focus on another, so I ended up developing a really complex project. Not long after I joined the lab and we started meeting regularly, I began to realize that something was very wrong.
Oh no.
I came to feel that the lab was completely dysfunctional and that my professor did not understand even basic science."
Been there.
Seems legit. He would give me incorrect advice, claim experience with techniques he clearly did not know how to use, and seemed unfamiliar with very basic anatomy, cell
Oh my God.
biology, chemistry, and even things like calculating inhaled dose. In one of my
Can I just say,
first,
sorry, before you go on, I just want to say I relate to this listener hard. We'll get into that in a
yeah,
minute.
yeah. In one of my first studies, we wasted precious samples on a method that had never even been validated. A lot of time, I felt like I was being used as a tool so that he and the postdoc could get information, protocols, and help from my committee members and other collaborators. The postdoc in the lab knew the professor before he started and has basically acted like his third eye for the past 5 years.
God.
There's
Okay.
literally so much I could say, but the situation became much worse after our annual meeting. I was very active there. I chaired a session, I was elected as a student representative, and I felt like I was finally building momentum in my career.
Love
Right
that.
after that, the professor suddenly seemed to shift the vision of his lab. After I shared what I wanted to do next to my career and mentioned possible collaborators that were connected to my final aim, he began using the postdocs project to establish those same collaborations.
Lordy
At the
Lord.
same time, he also seemed to be working to damage my reputation with one of my committee members. Now, there were other incidents that made me feel increasingly unsafe and targeted. Lots of things happened, but the one that stood out to me was about 10 days after I started the experiment. My cells developed severe contamination. I reported it immediately to both the professor and the postdoc, and I told them I would handle the decontamination, but I wanted to make them aware that something seemed to be actively contaminating the lab. What surprised
Mhm.
me most was how calm they were. They acted like it was nothing and just told me it happens. That reaction felt deeply off to me because my professor normally overreacts to much smaller issues, even things like doors being left open. After I decontaminated the lab, I then found out that someone had turned off the incubator, and around the same time, the alarm for the -20 freezer went off on a Sunday because the temperature had started to rise. Somehow, the postdoc happened to be in the lab at the same time these things were happening. The professor basically dismissed the whole thing by saying that a couple degree fluctuation wouldn't damage samples. To me, that makes no sense because without actual evidence, a temperature record, and a clear time curve, you can't really make that kind of claim. Plus, the temperature data did not even support what the postdoc had said. At this point, I feel completely certain that they're sabotaging my dissertation. I also want to add that the postdoc has threatened me several times over other issues, including saying that I would end up mastering out, along with other more serious comments. Whenever these things happen, the professor would either cover him or dismiss it as a joke. It only stopped once I requested not to participate in group meetings anymore. I'm dealing with what feels like serious bullying, manipulation, and intimidation, and there's so much more that I could say. What I regret most is that I listened to people who told me to stay in this lab even after another PhD student had already left because of their behavior. What should I do? Gosh, there's a lot to break down there, I feel.
There's a lot going on in the labs these days. What can we say? There is a lot going on in the lab. I feel really bad that they like regret staying, but also now, like at the very start, they said they started their PhD in 2021. That's 5 years, Taz. That's insane. 2021.
Crazy, but US is so long, I think.
It is long, but it does say they're close to graduation.
Yeah. So push
But
through is my initial thoughts. Definitely push through, try and finish if you can,
I know,
and
and I
then you
feel bad
can
to say
get
that
out
now
of
because
there.
they said they regretted listening to people who told them to stay in the lab. But I'm like, do you know what? If you've made it through all of this and you're like that close, I do feel like it would be a shame to give up now. But then at the same time, like, if you feel like you're being seriously bullied, then it's not an environment you want to be in. So my main thought goes to like, do you have enough data now to just leave the lab and just start writing up
Mm.
your thesis? Like, is any extra experiments that you are planning on doing really that necessary, or can you just leave the lab now?
Yeah, 100%. And regards to like your collaborations, like as much as it's lovely to have a supportive supervisor who will support your collaborations, it really does seem like he's going behind your back. You can be responsible for your own collaborations, honestly.
100%.
I think you should always take it into your own hands when
Yeah.
it comes to networking with other people and like your committee members and stuff like that. So definitely don't feel because your PI is trying to make those same connections, like you one have to facilitate that, and two can't make those connections yourself. So 100% you should get out there, start networking. Obviously if you're
Yeah.
at the end of your PhD, then you need to be making those connections anyway if you want to stay in academia. So yeah.
And we seem to have like an epidemic of arse-licking postdocs in academia because
Not just that, but supervisors that don't know how to do anything.
yes, and that's what I was going to say, is that them saying that they're dealing with a supervisor who doesn't seem to understand basic biology, like I have been there, and it's really tough to deal with that. Like I have been in a job before where I'm constantly having to explain the very basics of my field. And I understand I am the expert in this field that you've employed to advise, lead on these projects. However, I still don't expect to have to explain the very basic concepts of epigenetics every single time I meet with you. Like I genuinely, that was my life, and I found it really, really frustrating because I would explain a very basic concept and then it would be like, oh well, can we do this, can we do that? No, we can't do that because that violates the basic rules of epigenetics, so like we can't do that. And then 5 minutes later into the conversation, how about we do this? I just fucking told you that that's literally impossible. And I'm sorry to swear, but I do find it very irritating because I'm like, you've literally employed me because I am an expert in this field, and you don't seem to be listening to me. And again, we spoke about science communication on this thing. I don't think it was necessarily that I was bad explaining it. I think I was very good at explaining why what he wanted to do was quite literally impossible. But it felt like it was just falling on, what's the saying? Deaf
Deaf
ears
is.
every single time. And it's very, very frustrating because I feel as though you actually cannot have constructive conversations with your supervisor, and you don't actually get any help because they don't understand anything that's going on. And it's so frustrating for
Mm-hmm.
that to be like your everyday life. So I 100% get their frustration with that because it's very annoying.
Yeah, I honestly know a supervisor who like works in supervising people in the lab, and they have never done a day of cell culture in their entire life. And I'm like, how that is like the most basic skill in the lab to be able to do cell
I would never
culture.
do that.
How are you a supervisor of no, but it's wet lab stuff, so
Yeah,
I
yeah,
wouldn't necessarily expect you to do that, to know how to do that.
yeah.
But like, how are you a supervisor in a wet lab and you've never ever done cell culture? Like
Yeah, I
that
do.
honestly blows my mind.
But also, even if that is the case, right? So for like, yes, let's take this example. It's like, okay, you don't know how to do it, but like, can't you then collaborate with a PI who does know? So like, you don't have to be the sole supervisor of this project. Like, this is academia. No one's forcing you to supervise this project alone. Why don't you pull in someone who actually does know? Because every supervisor will play their part. Like that supervisor will be supervising that project because maybe they have other expertise. But like, if that's not your area of expertise, like pull in someone else. I always feel like, why do we only have one supervisor? I mean, I have always had several supervisors, minus that one that I just
Mm.
told you about. I've always worked on projects where I've had several supervisors, and it's always made so much sense to me because you need someone who has expertise in all the different areas to actually have constructive conversations and actually progress well within your project. Do you know what I mean? So
Mm.
yeah, basically I relate so hard to the whole them basically not knowing what's going on, and I understand why that is so frustrating. But yes, we do have an epidemic of nosy arse licking postdocs in labs recently, because I feel like we've heard so many so many stories about that.
Yeah, I mean, I don't know about the sabotaging in the lab situation. Obviously, we hear about it quite a lot on this podcast, but like, I think obviously just their actions alone when it comes to like the governance of your own PhD and also like how they're kind of sabotaging your connections, that is like warranted enough to be annoyed. But then I think when it comes to like, we've discussed it on the podcast before, like someone purposely contaminating your
Yeah,
cells, it's
so
just like,
often.
and to be fair, like, I don't know many people that take contamination like really seriously. I know they're very much like let's decontaminate the whole lab, but like honestly, if someone's taking charge of that, it's honestly one of those things. It's like, yeah, you go ahead, take that on. Like, I don't think it has to be like a full lab effort. So
Yeah.
yeah, but maybe
I wonder if
that's
it's just like, you know, when you have loads of like medium-sized issues and they all pile up to the point where you then take a small issue
yeah,
and blow it up into something bigger. We've all been there. Like, this is ultimately why, you know, like I had to leave one of my positions once because I found myself getting so worked up over like the smallest of things. But I think it was just because I was annoyed about so many other things that weren't being dealt with. So then, like, if someone sends me a CSV file and it's not got a correct name, I'm losing my shit. Like, I'm going to get really annoyed. And I was like, this is literally the end of the world. And I literally had to sit down with myself one day and be like, Liv, this is not the end of the world. You're now blowing this out of proportion, but it's because you're annoyed about all of these other things. If you can't sort these other things, then maybe it's just time to leave. I mean, ultimately, it's up to you what you do from here. I know you said that you were frustrated that like you had listened to people in the past and not left. If you were like a year or two in, I would give all the usual advice that we usually give, which is to number one, either leave, or number two, to go and talk to another PI in your at your university and get them to come on the project. But maybe because you're so close to the end, it is a case of just like getting on with it and trying to just get it done. But I would
mm,
say your next best option is to probably try and get in another PI or to just call it a day in the lab and be like, I've got enough
and
data, let me
finish.
just finish up here, you know?
Yeah, for sure.
But also, I'm kind of getting the vibe from this listener that they just wanted to like share their story and put their story out there to say as a message to like other younger PhD students who are going through a similar situation that they do regret not listening to other people in the lab. So I feel like they're also just trying to be like, do you know what? Sometimes if it's costing your piece, then it's not worth it and leave early. So I feel like
For
it's always
sure.
nice to hear people's experiences, as shit as they are sometimes, and get people's perspective on things, because we are always very quick to say like just continue. But if you are a younger PhD student, I feel like this listener is trying to tell you like it's okay to leave the PhD early if you feel like that's what you need to do, you know what I mean?
Yeah, for sure.
But definitely write in and let us know what you decide to do. Are you leaving the lab? Are you going to continue and get the PhD? We want to know how this ends up for you. But thank you so much for writing in and sharing your story. And if any of you guys are listening and you have a similar story, don't forget that you can head over to soculturedpodcast.com where you can submit your story anonymously and we will react to it on the show and give you our thoughts, feelings, advice, or just read it out on the show for other listeners to listen to your experience. But with that, that rounds off today's episode. So we will see you guys in next week's episode. Bye, everyone.
Bye.